Live data analyses are a common conservation paleobiology tool that compares living and death assemblages. A living assemblage represents the organisms currently living in a location. In contrast, a death assemblage is an accumulation of easily preservable remains of dead organisms from the living assemblage, such as clam and snail shells. Over time, dead remains from different years and generations become mixed together, resulting in a time average assemblage. This means that the organisms whose remains are preserved within a death assemblage may not have been alive at the same time. The gradual way death assemblages form result in a lag effect of species preservation, acting as a memory of what lived in the recent past. If a living assemblage changes, it will take time for the death assemblage to reflect that change, sometimes resulting in a mismatch. To confirm that the death assemblage samples are relevant to the research question at hand, it is important to radiocarbon date them. Radiocarbon dating not only determines how long ago organisms in the death assemblage died, but it also helps quantify the amount of time that is represented in the sample. By comparing species between living and death assemblages, conservation paleobiologists can determine if the community is changing over time and identify clues as to which potential environmental stressors, like habitat change, climate change, invasive species, overharvesting of wild species, and pollution, could be causing that change. For example, researchers at the Paleontological Research Institution, or PRI for short, are conducting a New York Sea grant-funded live dead study to assess the magnitude of the biological responses to recent reductions of pollution in Long Island Sound. Long Island Sound is the second largest estuary in the United States, extending from the New York City metropolitan area to the Connecticut-Rhode Island border. The Sound provides many benefits to local communities, including economic prosperity, habitat for wildlife, and recreational opportunities. Due to human activities, sources of excess nitrogen, including fertilizer, stormwater runoff, wastewater treatment discharge, and sewage outfalls, increased dramatically since the 1800s in the Sound. This influx of excess nitrogen results in local effects like harmful algal blooms and summer hypoxic events, or extreme drops in dissolved oxygen levels, which can harm the marine organisms as well as the overall health of the Sound ecosystem. In response, federal and state agencies developed a management plan to improve water quality by reducing point source nitrogen pollution by 58% and non-point sources by 10%. Ultimately, the management intervention proved successful, with the nitrogen reduction goals being met by 2017. Although water quality has improved, it is unknown if the marine life in the Sound has responded similarly over time. The PRI project compares living and death assemblages of mollusks, such as clams and snails, collected within the Sound to assess how the improvement in water quality has affected the molluscan communities, and how much more nitrogen reduction is needed to promote their health and continued survival. PRI researchers are identifying and counting thousands of shells, classifying each species' pollution tolerance. Radiocarbon age estimates indicate that the death assemblage samples date back to the late 1990s, making comparisons before and after the 2001 management intervention possible. Live dead analyses are one tool from a conservation paleobiologist toolbox to help extend the ecological baselines further into the past and identify potential environmental stressors that could be causing local change. Beyond Long Island Sound, live dead analyses can be applied as a conservation tool in other environments, such as rivers and lakes, and with other organisms, including tiny crustaceans like ostracods, and diatoms, which are algae with cell walls composed of silica. Additional sampling methods like coring can help provide even longer records of change in a given location to establish baselines from deeper in the past, such as decades to centuries to millennia. Such windows to the past are helping to empower resource managers to make informed decisions that protect and preserve the natural world for future generations.
Thank you.